No, yeah, I told him he's going live.
So we are live now.
Will disappear.
Hello and good evening, everyone. I am Mohua Maulik, and on behalf of NIBNG family, I welcome you all to today's talk, which is part of the lecture series on genomics of health and well-being. This lecture series is being organized by the National Institute of Biomedical Genomics, or NIBMG, as part of the celebrations for the 75th year of Indian independence, or Bharat Ki Azadi Ka Amrit Mahotsav, which is an initiative of the Government of India to commemorate 75 glorious years of progressive India. And it's my absolute honor to introduce today's speaker, Dr. Mitali Mukherjee, who is currently professor and head of Department of Bioscience and Bioengineering at IIT Jodhpur. My association with Dr. Mukherjee goes a long way back, actually when I had just passed my master's degree, and since then I've been very fortunate to work with her at different stages of my career, each of which gave me a very fresh perspective to science. What I find truly amazing about her is that she has never feared to come out of her comfort area and take up very challenging research initiatives and execute them successfully by building large research teams involving students and experts coming from different scientific backgrounds. So to formally introduce her, Dr. Mukherjee did her PhD in bacterial molecular genetics from Indian Institute of Science, Bangalore, following which she joined CSIR Institute of Genomics and Integrative Biology, New Delhi, where she had spent major part of her scientific career. And she was involved in setting up the key genomic initiatives in the area of rare genetic disorders, population genomics, functional genomics of repetitive sequences, about which she is going to talk today, and Genomics. She has been the convener of the Indian Genome Variation Consortium project, which provided the first comprehensive genetic landscape of the Indian populations. And under her leadership, her team has pioneered the area of genomics, which aimed to integrate Ayurveda with modern medicine through genomics. And she was the program director of CSIR's Genomics unit, Trisutra. At IIT Jodhpur at present, she is the head of the Department of Biosciences and Bioengineering, as I mentioned, and also she is an affiliate faculty at the School of Artificial Intelligence and Data Science. She is presently involved in developing Center of Excellence in Integrative Precision Health. Dr. Mukherjee is also coordinating a project in the Jodhpur City Knowledge Innovation Cluster called Thor Designs, with that aims to carry out an integrated study of the desert ecosystem to propel discoveries addressing health challenges in the arid regions. Dr. Mukherjee has numerous publications in several international peer-reviewed journals. She has been the recipient of a number of national awards, including the very prestigious Shanti Swarup Bhatnagar Award, along with many others. She is an elected fellow of the Indian Academy of Sciences and National Academy of Sciences India, and is a board member of the International Consortium of Human Phenomics. So with this brief introduction, I now request Dr. Mukherjee to kindly take over the platform. Thank you, ma'am.
Thank you so much, Moa, and for the generous introduction. And I would like to thank Professor Padma Majumdar Arindam and all to give me a chance to speak about the work which on allorepeats, and I primarily would like to put it in the perspective of the current exciting opportunities which this area provides, and it's been an area on which I've dealt for over 2 decades now, but in a kind of with a very small group, but it's been a very exciting journey so far. So I would like to share that. So I'll just start sharing my screen. Let me see. So is my slide visible?
Yeah, it's visible.
Okay. So good evening, everyone. And I think I put a a kind of a provocative title. It says as Alu leaps in evolution, and not a leap of faith now. And I'll try to, as I go through the presentation, try to convince you about this. So this is, I would also, if you people are anyway traveling to Jodhpur anytime, please visit our campus. It's very beautiful. And this is one of the iconic sculpture which has been put as now people take lots of selfies around this, and it just denotes the mind and the tree and all put together. So it's quite an interesting area, place. So so alus earlier were considered as junk DNA, and why it was because it was coined primarily by Crick as a selfish DNA, and also the field took a back seat on not trying to study this, because there were many people who cautioned that not only if you work on junk DNA, it would be intellectually sterile exercise, but also it might be futile, where it might not give anything. During those times, in 1986, there was a group in India with Professor Shashi Shekhar and Dr. Brahmachari, who were actually trying to study these repetitive sequences. And they were kind of proposing that these repeats might not be junk, because they could adopt different kinds of structure, and the structure variability could actually govern gene expression and evolution. Unfortunately, this had very few citations when compared to what Crick's citation of this being called as junk DNA, and that name stuck for maybe 2 decades, 3 decades or so. But things started changing in the 90s when people started making some small discoveries. They said that we now need to have a comprehensive nomenclature. So Jürgen Brosius was one such proponent, and he had a very interesting article written in PNAS. And also Professor Brahmachari's group in ISC was looking at many simple repeats and sequences which could adapt zDNA sequences, and how those could actually regulate gene expression. So there were lots of in vitro studies, because human telomeres, they formed unusual structures, and the school was primarily trying to see whether repeats could govern function, and that could then kind of tell how from DNA structure you could also get more such kind of information. And also when the human genome got sequenced, there was a lot of betting amongst people as to how many genes humans would have. If we look at Drosophila, we look at grasses, there were lots of paradox by that time, something which was called as a K-value paradox, that if we say humans are complex, if we think that chromosome numbers are what gives complexity, that doesn't seem to be true. If we think about whether the DNA amount would make us more complex, that also is not so true, because amoeba, which to us seems to be relatively simple, has more amount of DNA in them than us. Then the human genome came out, and what was found was that the number of genes, as we would annotate, had not changed. So if a Drosophila or a grass or a human were to be compared at the level of gene numbers, there wasn't much difference. But what came out very striking was this interspersed repeat content, which is so far was thought of as junk. That number has steadily increased in the human lineage. So this red one is the human, somehow the label has gone off. So if we look at the human genome, there is a 50% of the genome which is non-repetitive, but 50% of the genome comprises of different kinds of repeats. And one major class are the retrotransposons. That means they jump around in the genome through a RNA-based mechanism. They make RNA copies which gets converted to DNA, and then they mix in the DNA, and they move to places where the nicks are produced, and that way their numbers increases in the genome if they are allowed to be transcribed. So amongst these repeats, there are different kinds of retrotransposons, and this ALU, which I would be talking about, belongs to the sine family of repeats. And the lines in terms of amount of space it occupies is much more. Sines or the ALUs occupy around 10% of the genome, but in terms of numbers, they are immense. They are nearly in 1 million copies in the human genome. And if we look at the expressing part of the genome, so if this is the genome and this is the transcriptome, the amount of transcripts, the proportion is much more with respect to ALUs. So this is the most transcribing part amongst the repeats in the genome. Typically, an ALU, it's around 280 to 300 base pairs in length. It is formed by actually two blocks of DNA, which have come from so it's called left arm and the right arm, which has come from fusion of two 7-SL RNA and the right arm has further 31 nucleotide insertion, and in the middle there is a poly A tail. What's interesting is it has got this A box, B box, which is a Pol3 promoter. So it's an internal promoter, just like the tRNA and rRNA, and it can transcribe itself and make ALU RNA by transcribing by Pol3. It has been, because its size is 280 base pairs in length. The studying them was much difficult in the pre-NGS era. And as the NGS publications have started increasing from 2010, the number of papers which are coming out where ALU is implicated is actually on the rise, and that's because as you go through, you will realize that since these sequences, though they are repetitive, it's very difficult to anchor them to a genomic region unless you have junction sequences. So studying them becomes very difficult. Now, we can very recently a bioarchive paper came. I am yet to see it in a publication, but it has been covered by a large number of prominent journals itself with the bioarchive citation. So that they talked about jumping genes may have erased tails in humans and other apes and boosted our risk for birth defects. It's come from a very prominent lab, IIT, and what is very interesting is they have actually tried to understand whether they could find the answers to the loss of tail in the lineage of the hominoids, and what they found by doing analysis of the genome was that all the animals which have presence of tail have this gene called TBXT, which is of a particular length. And in the hominoid lineage, there has been an insertion of an ALU repeat, which actually gives rise to an alternatively spliced protein isoform, which is smaller in length, because there is a loss of an exon. And apparently, during evolution, it has accumulated a number of mutations to be able to contribute to this loss of tail. Now, they went a little further and actually using CRISPR-Cas method, they made mutants in the mice. And the homozygous were not viable, but when they made heterozygous mutations, they found that the length of the tail were very varied. And this was one of the evidence which they showed that because of an ALU insertion, you can actually contribute to such a drastic phenotype as a loss of tail in the lineage. Now, we've started this journey way back in 1999 when chromosome 22 came, and all the people who might have interacted with Dr. Brahmachari, he was a very, he wanted to start genomics in a big way, and he wanted to see whether we could actually contribute to computational genomic space. So when the first chromosome 22 came out, he asked everybody in the lab to look at various aspects in chromosome 22. So I started my journey from here, and this is a very, very ancient kind of a map which one of my students, who also was not an informatics student, drew. We started to see whether we can actually start to visualizing these repeats in the genome. And when we did this exercise, we found something very interesting. So this is 1 MB or 1 page of the 20 of the chromosome 22, and this was because this was the smallest chromosome, and something actually made us kind of think whether there could be something happening with respect to allorepeats distribution in the genome. So there are these lines, and you can see these lines are all about their genes, pseudogenes, predicted genes, because 99 not too much annotations were available, and these all the vertical lines are allorepeats. What became evident was that we found variable distribution of allorepeats in amongst the genes of different sizes. There were small genes with more allele, less genes, larger size, again with not a proportionate increase in allele. So we started asking a question: is the distribution random? And this is where Professor Partha Majumdar actually helped us. This was, I'm not getting into the detail because it's an old story, but this was the first time when we actually read up all the chromosomes genes of 22, as well as by the time we could do the reading, because there was no tool like gene ontology available. So I and a student of mine, we sat with a biochemistry book and started classifying all the genes in chromosome 22 into these broad categories. And then when we looked at allele density in these genes, we found that there were certain categories which had more allele density and some which had very less allele density. And by the time the chromosome 21 came, it also showed the same pattern. And then we approached Professor Majumdar whether he could help us out to see whether there is actually a significant difference amongst the groups, and that's how we did some analysis. We did a lot of regression with respect to size, GC content, and all. And nevertheless, our results showed that the distribution of allorepeats in the genome is non-random. During this time, it took us nearly 2 years to get this paper published because it was considered as junk DNA. Nobody was concentrating on them, and we thought that we had something going over there. By the time the entire genome got sequenced, and when we had the first draft, we actually again looked at the patterns, and we found similar kind of patterns. And we were lucky during that time that we got the gene ontology tool out by Soren Dragici, and we could actually do an ontology, and we found that there were certain processes which like apoptosis and ubiquitination and all those kinds of processes in which alleles were very, very abundant, whereas there were certain processes where alleles were excluded out. So we started asking a question: is there a reason because of which alleles are more present in the proximity of certain kinds of genes? So another PhD student of mine, we started looking at regulatory motives. During that time, people were characterizing one gene at a time, and they were encountering that alleles have certain transcription factor binding site which could act as enhancers. So what we did was we took all those motifs which were reported, and this student Ravi Shankar, who is a faculty in IHBT Palampur now, he made some kind of motif search, which was based on hidden Markov model, and also because these are very small motifs, so they needed to be normalized by their background information, etc. But then when we did this study, we found something very interesting. We found that a large number of transcription factor binding site clustered within allorepeats, and this clustering of these allorepeats having these clusters of regulatory elements were also non-randomly present. So if there were genes of signaling pathway, they tended to retain more alleles in the promoter proximal region with certain kinds of transcription factor binding sites. Now, this was very interesting because alleles are transcribed by RNA pol 3. And here we find that there is a clustering of RNA pol 2 regulatory elements. So we tried to ask a question whether, because of retrotransposition, is there a possibility that allele with such kind of transcriptional motif could go on distributing such kind of regulatory motifs in other kinds of genes, and by that way, could there be a rewired network from the same set of genes? We actually this was just a computational biology paper. There were no functional studies, but we just sort of proposed that maybe because during that time there was specially a tool made which is called as repeat masker. The purpose of repeat masker was you delete out the repeats, and because they kind of complicate the work, and study the remaining part. And what we did was we used the repeat masker data and study them. So we sort of propose that if there are candidate genes which predisposes to diseases, and having such kind of containing regulatory motif might have physiological consequences. This was way back in 2004, but this is 10 years down the line. There are lots of papers now coming up. This is one such paper, but there are many in the literature where they talk about how allele elements can actually give rights to newly inserted alleles by accumulating mutation can actually become enhancers. So as they age, they become more kind of older alleles can serve as enhancers. And very, very recently in a genome research paper, more than 75 transcription factor binding sites have been functionally shown to map to allele repeats. So this was just a correlation for us. So we wanted to know whether it actually has a function. So one of the things we thought was that since alleles transcribe and they can make an RNA, is there a possibility that having a Pol2 regulatory element, can they, in response to a Pol2 signal, make a transcript? And supposing that transcript were to be made in an antisense orientation, because alleles are also exonized within the transcript isoform, would they kill the transcript? So this is one such experiment we attempted. So in our earlier motif search, we found that heat shock factor binding sites are there in alleles, and nobody had at that time shown a function of those binding sites in there. So what we did was we did a simple experiment. We treated HeLa cells with heat shock and looked at differentially expressed genes, and looked at alleles in the vicinity of those differentially expressed genes. And we found that lots of genes which are downregulated in heat shock harbored an allele element in the downstream region, which also contained a heat shock factor binding site. So we asked a question whether in response to heat shock, it could make antisense transcript. So lots of experiments were done, and including first of all trying to see whether we are able to locate antisense transcript, doing chip binding, and looking at whether these could bind heat shock, and then giving an siRNA in a heat shock cell against the antisense transcript and see whether we could alleviate the expression. So this way we could actually show that alleles harboring regulatory elements could actually make antisense transcript, which could then titrate out the sense transcript, and as a result of which you could have a downregulation of expression. So this again then demonstrated that if there are so many transcription factor binding site motifs in allele, then there could be so many ways it could ectopically get expressed. During that time, more and more functions were getting ascribed to alleles. So if you look at a particular typical gene, alleles were found in 5' UTR, they were found in exonization. There was a science paper which they said that alleles have 19 cryptic splice sites, and so as a result of which they can form a part of a transcript isoform. And then they were in UTR. What was also observed that large number of antisense transcripts also map to allele repeats. And a third thing which was serendipitously found by people was that there is a widespread editing of transcripts, that is A to I editing, which can actually change the base, meaning the coding potential or the translational potential of a transcript. And that 90% of them happen in allele, because allele form a particular kind of a secondary structure, which is a very good substrate for the editing enzyme, as a result of which large number of transcripts, if they have alleles in them And if ADAR1 or ApoBec are expressed, then they could modify those transcripts. So then we asked a very simple question. We asked that supposing there is a gene with a transcript isoform which has an alloexonization. So it could have a possibility that it is a transcript without an allo. It could have transcript with an allo, and it could have a transcript which is edited. And there could be a condition in which an allo antisense is also made. Then supposing if these, depending on what kind of species would be there, would you have different fates from the same cell? So then during that time, NGS data was not available, and we scrounged the pretty much all parts of all kinds of resources. For example, human RefSeq database to look at all exonized transcript and map them to different regions. We looked at different kinds of 11 million ESTs, looked at the SAGE data sets, and tried to identify if we could get allo antisense within them. And then also looked at editing by mapping to the genomic regions, and also ruling out the possibility that it could be an SNP variation, whatever amount of SNP data was available. So we did this because we wanted to know that if this were to crosstalk, then it has to be the same allo which has an antisense and an editing. If it is a different allo which is exonized, different allo which has an antisense, doesn't make a sense. So then when we had found all these kinds of data, we tried to look at whether these could interact at a exon level, and whether they are enriched in a particular process, or could they then modulate a particular regulatory network. When we did this, we actually found there were around 319 genes, mostly to DNA damage, checkpoint, cell cycle checkpoint, nucleoside, nucleotide processes and all, which had all these possibilities. So this was again a big correlation study, and we wanted to know whether this actually could happen. So luckily for us, there was this first of the RNA-seq in a temporal data had got available of this person called Michael Snyder. What he had done was he had sequenced himself, his RNA, in a very deep strand-specific manner throughout the year, and he had 2 viral infections during that time. And the literature had suggested that during viral infections, the levels of allo-RNA increases. So what we did was, so there were these 5 time course RNA-seq data set, and concurrently there was small RNA-seq also from there. So we fished out those sequences, and at different time points looked at the exonized transcript and looked at the antisense transcript. What we found was that right after infection, the exonized transcripts were lower in number as the antisense RNA was more, allo RNA was more, and as the virus infection tapered down, the antisense form containing the allos were much more higher in the genome. And when we looked at it, we found that there were around 60 genes which could be altered in such a manner, and they seemed to be linked to this pathway, which was ubiquitination link pathway. So this was interesting result, where now we show that if you have these modules of allos in a transcript, and if they are differently expressed during different times, then you can actually have a different outcome from the same gene, and it increases the repertoire of the things which could be there in the transcript. Over the period of years, now people are talking about allo elements and how they could actually be involved in the evolution of human brain and spectrum of neurological disorders, because all these kinds of processes have now been mapped to large number of genes, and they impact many processes which are key to ROS and mitochondrial and lipid metabolism, etc. And they could be playing a major role in many diseases. So if you look at the entire database now, or the PubMed now, you will find large number of papers where allo has been functionally shown. So then we started asking questions whether allos could actually accept and make more important contribution in the long non-coding RNA, because that's also gaining a lot of credence, and it is actually one of the important biomarkers. And then when we asked the first question from could ALU, if they get exonized in the 3' UTR, because it makes particularly secondary structure, could they actually be targets for miRNA? In that case, a new ALU getting integrated in 3' UTR of a region can actually get into a regulatory network of a miRNA. So we again did a very simple experiment in a HeLa cell where we gave a heat shock to HeLa and then looked at the transcript. Then we looked at miRNA, which are differentially expressed, then tried to do an informatic and then functional genomic analysis to be able to see whether these miRNA, which are conserved miRNA, could actually interact with miRNA binding sites in ALU, and could that actually govern differential outcome of transcripts. So and then what we also did was we overlaid it with variations which map to these miRNA targets, and we could also look at that there are some variants which are under positive selection reported in the literature. Some of these were present in these ALUs containing these miRNA binding sites, thus kind of suggesting that such kind of elements could be functional. miRNA literature most of the time has grown by comparative genomic analysis, but if something is primate specific, then there's no way we can actually do a traditional way of comparative genomics. So we'll have to look at between individual in a population specific manner. So that's where some of the learnings from the Indian genome variation came in useful. But during that time, a student of mine actually found a very, very unique transcript, which we call as ALU-LT. LT is the large transcript of a particular isoform of in a gene, which is very, very conserved across evolution. This gene is called as CYP20A1. And what we observed was that it had one long UTR, which was over 8.9 kb. And there was a exon which was skipped in this, and this one, if we look at the entire 3' UTR compendium, is amongst one of the largest UTR. It is actually in the outlier region. What was very interesting was that because we could distinguish it with respect to one exonic region, we could use junction sequence and blast the RNA-seq database, and we found that it's expressed in many cell lines, including some cortical neurons in the Allen brain atlas in a unique neuron, which are rose hip neurons, which are unique to humans. Then this was followed up by various kinds of analysis. What we found was at the protein level, it was very conserved, but this UTR seemed to be unique to human and was very, very sparse in other genomes. Then what was very interesting was that when we looked at this 3' UTR, 85% of the 3' UTR were made by 23 ALU repeats, 23 different ALU repeats. And what was most interesting was that it gave rise to around 4,700 MRE sites of 1,000 different kinds of miRNA. Now, we thought that because it's a repeat sequence, it could well have come from all these repeats being together. So we did a randomization and we looked at 10,000 such sets of 23 ALUs, but we could not find such a dense distribution of miRNA binding site, which suggested that these sites could have originated after they were exonized. Now, when we mapped them, we found that there were some miRNA which had as high as 43 numbers, 20 of them. Most of them had 20 sites, some are 10. So this kind of striking distribution, and they were mapping to different kinds of ALU subfamilies. So these are 23 ALU subfamilies, and these are the miRNAs which had more than 43 sites, more than 20 sites, more than 10 sites. So this is in that long transcript. We did race and all, and we did sequencing to confirm that this transcript is really there. We looked at its localization, all that was done. And then we asked a question, if this has so many miRNA binding site, could that act as a sponge? So that is what we tested. Our hypothesis was that if this were to be acting as a sponge, then whatever miRNA are expressed, if this titrates it away, then the genes which harbor these sites would be upregulated. So there should be a positive correlation with when the CYP is expressed, the targets should also be expressed. And if this is down, then the miRNA are free and it can go and kind of regulate the transcript. So then we had to find a condition, a similar condition in which these are oppositely regulated. Luckily for us, we could approach Dr. Pankaj Seth in NBRC, who has this TAT model, and we found that in HIV TAT treatment of primary neuronal cells, this expression of gene is up, and if heat shock is given to the same cells, then the expression of this is down. So this gave us a window to test our hypothesis, and we did an RNA-seq and strand-specific, and we also looked at the miRNA expression by sequencing, and we found that 9 miRNA, which had more than 10 sites in that CYP20A1, were expressed in that, and there was a correlated expression of around 380 genes which harbor the miRNA target sites. So we looked at whether the mean target sites were more in the differentially expressed by comparing it along with a random set of genes. And every time we found that the 380 genes which had those miRNA sites had more numbers compared to a random set of genes. Most interestingly, we also found a correlation with these 380 expressed genes with the CYP20A1. If we did it with all the differentially expressed gene, we did not see such a correlation. Then when we looked at these gene sets, which are 380, which are actually co-regulated by this, we found interestingly certain processes which I'd never heard of, blood coagulation in a neuronal gene, and the neuron development, mRNA processing, and all those kinds of things. What we then looked at the literature, and we found that there is some newer evidence coming up in neuroinflammation condition where this kind of a phenomena is reported, and there is a possibility that such kind of long non-coding RNA, which have such uniqueness of a sponge, could actually be maybe important in certain neuronal functions in the genome. So this was again not directly tested during that time. So then we decided to test it, whether by cloning the 3' UTR fragment, because it is very difficult to test it in the same genomic location. So we cloned the fragments of the CYP20A1 with all those miRNA binding sites, and our hypothesis was if this were to be expressed, and if those miRNA are there, the expression of renila would be going down. So we tested this. We actually had the different clones, and we could find that if the activity of reporter is decreased when you have different fragments containing CYP20A1, with respect to the miRNA which are expressed in the SKNH cell line. We also did this pull down of AGO, which would tell that the RNA is bound with miRNA, and we could found in the IPSA that was binding to that is associated with the risk complex. And then what we did was we looked at the target genes from our data set, that if we transfect it in that cell line, what happens if the clone is overexpressed, that is, it is titrating out the miRNA, then the expression of the genes in which they are target, they get overexpressed. So the opposite one, if we knock down this with siRNA, then opposite should happen, and that's what we could actually demonstrate. This was again in an in vitro cell line. So this GAP43, important neuronal gene, the clone has these many sites of these miRNA. CYP20A1 has these many because we could not clone the entire fragment, and we could actually see a finite decrease in expression in CYP20A1 expression. So this kind of showed that if such a kind of a phenomena, meaning is possible to happen, and it again adds a new dimensionality in terms of a particular environmental insult or in some way by which the CYP20A1 get induced, then it might also be titrating out a large number of miRNA through such kind of a sponge mechanism. Alus are now being shown to be a large fraction of circular RNAs in liquid biopsy, and such kind of a sponge effect of alus are being mentioned. And the last part is, so these alus, when we are talking about, are stable alus. But then what happens if alus are showing insertion deletion polymorphism? Insertion deletion polymorphisms means an alu is newly inserted, so the other allele is called as a deletion allele, and the one which has the insertion, new insertion, is called as the insertion allele. Such kind of polymorphisms have been studied extensively. For example, angiotensin converting enzyme, enormously important during COVID time. It has a insertion deletion which is related to angiotensin converting enzymes levels, and it also has been the insertion deletion polymorphism has been linked to phenomenon of high altitude adaptation, physical performance, etc. So alus as population markers, ancestry markers are known, but now it is also, if this kind of gets inserted, deleted in a individual specific manner, then you can realize that because of its various kinds of functions, it could bring about different networks, right? So there were reports of structural variations in world populations. In India, very few studies were there, but then we decided that when the indigene came, we decided, could we actually start to see whether Indian populations have new insertion deletion polymorphism, and whether those could actually be useful as ancestry specific marker? Are they different from world population? Do we harbor new alu repeats, and do we need to be concerned about them? So we looked at 1021 Indian genomes, experimentally validated, and we found 90% concordance with a large number of sites which we found insertion deletions were there. And then we looked at their patterns of distribution, compared it with global set, and also looked at in which kind of genes they are more prevalent. So we found lots of alu insertions, and predominantly they were of the alu-Y subfamily, which are the ones which are actively retrotransposing at this point of time. And many of them were common. A large fraction was rare also, and many were private. On an average, one individual can have 770 insertion deletion, which were different from the reference genome. A large number of these jumps happened in the genic region, which again would seem to be counterintuitive, but this is what we have been seeing. Alu distribution is non-random and more present in the genic region, more in the intronic and also in the 3' UTR. And when we compare it with other data sets, for example, South Asian and in the 1,000 genomes and all, all of them, we took the ones which had a reasonable same coverage. We found that there was an intersection of insertion deletion which was shared by all population, but a large number of them were also unique to indigen. And a common one, meaning there were 7 of them which were unique to other population, which was common in India, and others were actually, a lot of them were common. And this is how the frequency distribution you can see between the populations. So some were rare in other population, has become common here. Some common has become rare here. So we got all kinds of distributions, but 60% of them are unique to indigen data. When then we looked at can we use them as ancestry informative marker, we took all the shared indels, and we could find. So this is the indigen samples. These are European samples. These are East Asian samples. These are African samples, and these are the ones which are Indian. And then we took the indels which had high FST, and we could actually find similar kinds of distinguishing things. So which tells us that this kind of a panel of indels would be very useful in and population genomic studies also. Then we looked at which are the processes in which alleles tend to jump. And again, we found something interesting, that processes related to neurogenesis, cell morphogenesis, and all of them are very, very enriched in newer jumps. And then we try to intersect it with the neurodegenerative disease variation database. We also found that large number of these variations, allele indels, were there in the genes which are implicated in neurodegenerative disorders. So this part, we could say that if we, when we sequence these genomes, we identified a whole lot of indel polymorphisms which had a bias distribution. They could be serve as utility in ancestry, as well as they could have potential biological implications. So this we recently published, and this is a very big compendium, and the entire link is present in the ALU database. So just to summarize, now you can see ALUs have a role at the genome level. More and more work is showing that it also has an effect at the epigenome level. At the transcriptome level, it can have various kinds of forms having a function. And even at the proteome level, people have detected short peptides which are only comprising of ALU or newer exons derived from ALU repeats. These have all been many studies, but they are all very disconnected, meaning people are not looking at whether in an integrative manner could they actually govern outcome. But one thing which is coming as very common is that a stress, it could be viral, bacterial, heat, UV, hormone, DNA damage, all of them induce these RNA, ALU RNA transcripts of different kinds. And these all processes govern different kinds of cellular responses, which have these different states of the cell, either differentiation, apoptosis, or proliferation, and they have systemic impact. So ALUs are now important key players in inflammation, in fibrosis, and all. And each of these are relevant in many complex disorders. So this kinds of tells that this cannot be ignored anymore. The last 3 years have actually seen a phenomenal increase in the various kinds of studies which have reported. A very unique paper I saw has come where they actually talk about ALUs as flip-ons, that is sequences that form non-BDNA conformation in physiological, and they say T flip-ons, quadruplex, Z flip-ons, based on what kind of structures they form. And so an ALU element present in a transcript by an epigenetic modification can form a secondary structure, and the secondary structure can be of any of these kinds, and each of them has a different effect on gene expression. So this actually tells that it adds more to the regulatory network by even having a structure as a information component in them. There is also been a study where they have shown that presence of ALUs in the upstream region were actually define boundaries in a tissue specific manner, and could govern actually epigenetic environmental response. Many other new things are coming up. ALU in long non-coding RNA can, through their motif, bind to DNA in a triple helix manner and bring the entire chromatin remodeling machinery. And this has also been shown to be involved in atherogenic conditions. So such kind of examples are coming up many times. So now where are we headed? So this is the last part, that when I moved to IIT, and this requires a huge amount of computational analysis looking at motifs. So I started a program here. It is a continuation of what I had been doing in a cottage industry kind of a manner. That can we actually make a database which is like a kaleidoscope of value-mediated regulatory transactions. So if people actually have any data set of their choice, if they put it in such a kind of a database, they should be able to know whether ALU could have an involvement and in what functional hierarchy. So the idea is to create a compendium of all these data sets from different experimental platforms to be able to do integrative analysis on the fly. So this required different kinds of skill sets, which I do not possess. So here I've tied up with a colleague, Deep Shankar Banerjee, who works on AI and AI architecture, big data architecture, Ishan from IIT Delhi, or both of them computational students. Prakriti, she was with me and then joined Ishan as a computational biologist. And Deep, he's a second year BTech student from computational sciences. He's got very charged and he's working with me the last 6 months and trying to see whether we can actually build a team over here who could study these. So this is the final acknowledgement slide. It has been a long journey of 2 decades, but I've done it. One student has developed one model or one observation, key observation, and a wonderful set of collaborators and the Indie genome. Like to thank, and thank you all for listening to this lecture. So I'll stop sharing and I can take questions if there's any.
Thank you, Dr. Mukherjee, for this absolutely fascinating talk. It's really very interesting to see how this field has evolved and how we have progressed with this. And so, Alpana, can you please check if there are some questions in the YouTube chat box?
Not right now.
Not right now. Okay. I'm really intrigued to know about those CYP20A1 LTs about in coagulation pathways, right? In the neuronal cells. So yeah, if you can just briefly talk about that a little bit more. I'm very interested to know about that, like how they are associated with different kinds of neurodegenerative conditions.
So I do not know whether they are really, meaning we found functional enrichment of those genes, and those genes upregulation has been shown to be involved in system sclerosis and other diseases where there is this breach of the blood-brain barrier. So when the coagulopathy came, so Aniket said that I got a kind of a enrichment which doesn't make sense in the neuron. So then we started reading up, and then we found that this is a whole new term which has come up, where they are talking about this aspect. And we found that these miRNAs target the set of genes which are all involved in this pathway. So very interesting is that introduction of a sponge kind of a sequence can really make the system go very cooperative. If you have one miRNA mapping to every gene in the genome, then the cell will be behaving in a very non-intelligent energy manner, right? But if you actually have a sponge which can totally titrate away all the miRNA from related set of genes which have to participate together, and also release them when it is needed to be shut down in a manner, then this kind of thing could be actually involved in rapid responses in a regulatory network, and maybe addition of such kind of motifs could, to the same set of genes which are otherwise conserved across evolution, could be making the processes more complex, right? And there are 1000 miRNA binding sites. So just like transcription factor, which are housekeeping, meaning genes which are housekeeping, accumulate all kinds of transcription factors in them, right? Because they have to be expressed in all tissues. So also these miRNA, so many binding sites must be a need for it to be switched off in most of the cells, and only when there is a need it can maybe switch on. As an inflammatory kind of a viral infection or whatever it is, it needs to be tested.
And also there can be some age related changes, right?
Possibilities there. So we were just curious because, as you know, I do all kinds of these analysis, and after that I get stuck in functional studies. So I reach out to different people. So we reached out to the Allen Brain Atlas people to see whether if this is a long non-coding RNA, it should be uniquely present in certain brain regions, and they were very forthcoming. And just because we had a small sequence which could differentiate, they could rapidly look at 16,000 single cells in the rosehip neurons and show that this is expressed. So, yes.
Absolutely fascinating work. Very interesting. Thank you so much.
Yeah. Yeah.
So any questions from the house or in the YouTube? Alpana?
No, nothing on YouTube yet.
Nothing on YouTube. Okay. So if there are no more questions, then I would like to thank Dr. Mukherjee once again for accepting.
Yeah, Mohua, before you thank, let me ask a quick question. Mitali, these particular regions of the genome where there are these allorepeats, has somebody more recently done any selection analysis, whether these are under purifying selection, advantageous selection, do you know?
So there was a work by King Jordan. Also, some amount of selection work has been done, sir. I can pass on the paper to you. So,
Okay, I would be
because
interested in
these
that.
essentially these insertion deletion polymorphisms have been shown to be in LD with a large number of variations, which have some of them display positive selection. So, like those kinds of studies have been done. So nowadays, the last 3 years, I have not read extensively, and I was from when I was preparing for this, I find that anything I search, and I find that there have been people who have addressed some questions or the other. So I'll get them across to you. We lost him. I think he lost.
I think he froze. His window froze.
Mm-hmm. I think so.
Yeah. Okay. So yeah, thank you, ma'am. Thank you very much for this wonderful talk. And we actually would have really loved to have you on campus, but unfortunately, we have to do this online this time.
Yeah. If I'm
Maybe
coming
sometime.
there, yeah.
Yeah. We would have loved to have you here.
Yeah, surely, Mitali, the next time you have to be in NIBMG. Let me pitch in here.
Yeah.
Yeah.
So thank you so much.
There's a huge amount of, so I'm kind of seeking more and more people to participate in ALU. I want to do it as a crowdsource kind of an initiative where people from their domain knowledge can contribute, or we can look at different data sets to make better models to see whether there is an involvement of ALU in any of these events. And the BTech students from computational sciences and others are very charged. So we have design credits from second year. They can actually take up a project. And sometimes you give these projects, they are very interested in doing computational analysis. So trying to build up a team here. And anybody would like to pitch in or participate, more than welcome.
Sure. So,
Okay.
yeah, thank you. And with this, I also thank all the participants who have joined us today online and our IT team for the support and making this event a success. And so we come to the end of this event today now. Thank you, ma'am, once again, and thank you, everyone. Bye-bye.
Thank you. Thanks.